Bismillahirrahmanirrahim. 5090 November 24 question paper 2-1, and we'll try and do the first few questions on that. Question number 1, figure 1.1 shows an experiment to measure the water loss from a small plant in 24 hours. There's a measuring cylinder, there's a small plant, layer of oil on the surface of the water, there's water, and there's a top pan balance, and you can see there's some sort of a digital reading that we can see. Name the process by which a plant loses water from cells to the atmosphere. Transpiration. Part 2 suggests the purpose of the layer of oil. Now you see, in another experiment, the layer of oil would be preventing oxygen to allow anaerobic respiration, but here it is preventing water loss by evaporation. Then part 3 of the question describes one way in which a student could use this apparatus to obtain a value for the amount of water the plant loses in 24 hours. Well, you measure the volume at the start and at the end of 24 hours, and you subtract the end value from the start value, or you calculate the difference. So measure volume at the start and the end of 24 hours and then calculate the difference. Then coming to the B part of the question, water is lost mainly through the plant's stomata. Name 3 types of cells that the water passes through before it reaches the stomata and the leaves. Write the names of the types of cells in the correct order in the boxes. So, well, of course, the first one would be the root hair cell, because you see the water from the soil enters the plant through the root hair cell. So, root hair cell, then you can also say root cortex or cortex cells. And then the xylem vessel, and then the palisade or the spongy mesophyll. After this, so then of course it will be. So you could have given me any of these. After this, if you could have also written palisade. So anything in the correct sequence. So you could have said, instead of root hair cell, you could have said root cell or root cortex cell, and then xylem, and then palisade, mesophyll, and then of course stomata. Then part C of the question. The experiment shown in figure 1.1 was repeated many times to investigate the effect of humidity on the rate of water loss. All environmental conditions were kept constant except for humidity, which was gradually increased. Shows the axis for the graph. So the humidity is increasing in this direction and the rate of water loss is increasing in this direction. So when there's low humidity, say it's 1 here and it's 5 here, in low humidity, then there'll be more water loss. So you go to graph like this. At low humidity, what you're seeing is that this is the leaf, and inside we have say 100, and outside we have 10, so the gradient is very steep, so there's going to be a lot of transpiration or water loss. But as humidity increases, say it goes up to 100, well then of course there's going to be no transpiration. But even if it is 90, the humidity, you go to say we are increasing the humidity. So initially the humidity was say 10, and then it goes up to 90 outside the leaf, and it's 100 inside the leaf. So you realize that if you increase the humidity, then transpiration, if the humidity outside is say 200, then increasing the humidity will not increase the water losses. So inside is 100, outside humidity is 200. So there's going to be no water loss. So more the humidity, less the water loss. Part 1 said sketch a graph on these axes of the expected result of this investigation. Now it says explain what your graph shows. Explain what your graph shows about the effect of humidity on water loss. And has 3 marks. The lower the humidity, the higher the rate of water loss. Water is lost by diffusion. When humidity is low, the concentration of water outside will be lower than inside the leaf, and atmosphere would not be saturated with water. So low humidity, high water loss, water is lost by diffusion. In low humidity, the concentration of water outside will be less than inside the leaf. Question number 2. Maltase is an enzyme found in the intestine of vertebrates, including humans. Consider table 2.1, where it provides description of words related to the activity of maltase. The first row has been completed for you. Maltase, an enzyme that is found in vertebrate intestine. The substrate of maltase, maltose, glucose, product. It is a product or what is produced when maltose is broken down. Then part of the maltase molecule which fits the substrate. Active site. Then specific. Maltase only acts on maltose. Then coming to the B part of the question, vampire bats live in South America and feed entirely on blood. They are the only known vertebrates which do not have maltase in their intestine. Suggest an explanation for this. Well, why? Because there's no maltase and there's no maltose in the blood. Blood does not contain maltose; it contains glucose. So blood of they feed entirely on blood. They're vampire bats. So blood only contains glucose. The other one, of course, you could also say is maltase not produced. They do not waste energy producing an unnecessary enzyme. Maybe it is a result of mutation and evolution. And over a period of time, this does not have a functional gene for maltase. No maltose in blood. Maltase not produced by these vampire bats, as do not waste energy producing an unnecessary enzyme. Mutation could be a result of mutation and evolution, and does not have functional gene for maltase. Question number 3, figure 3.1 shows the central nervous system and some other parts of the man's body. Name the two parts of the central nervous system labeled A and B. So A is the brain and B is the spinal cord. So as you can see, A is the brain and B is the spinal cord. Then coming to the B part of the question, it says the man accidentally touches his hand on a sharp pin and immediately lifts his lower arm away from the pin in a reflex action. The reflex action involves a receptor and an effector. The receptor on figure 3.1, draw a label line to the location of the receptor and label it R, and effector label it E. So receptor would be in the hand, so this would be R, and the effector would be the muscle, so this would be E. So effector and receptor, and you have to go a label line. So to the hand it will be the receptor, which is skin has got receptors. So we have this arrow pointing to the hand, and E would be the effector in which the muscle contracts and you withdraw your hand from the pin. Then coming to part 2 of the question, describe the roles of two named neurons in this reflex arc. In a reflex arc, there are three neurons which we talk about: sensory neuron, relay neuron, and motor neuron. So for 4 marks, you just give me all the points about these three neurons. So sensory neuron receives impulses, is stimulated by the receptor, carries impulses to the spinal cord, or you could say the central nervous system. Then the relay neuron carries impulses from the sensory neuron to the motor neuron. It sort of connects the sensory and the motor neuron. And the motor neuron carries impulses from the relay neuron, or you can say the spinal cord, or the central nervous system, to the effector, which can be a muscle or a gland. Here, of course, it's a muscle. but it can be a gland as well. So as you can see all this how it is worded, sensory neuron is stimulated by the receptor, carries impulses to spinal cord. Relay neuron carries impulses from sensory neuron to motor neuron. That's in fact 2 marks. And motor neuron carries impulses from spinal cord or the central nervous system to the effector or the muscle. The part of the body labeled C is a gland in the endocrine system. Name gland C. You can see that this is, of course, everybody knows this, I'm sure, is just above the kidneys, and this is called the adrenal gland. Renal is for kidneys, so adrenal gland. So this is be the adrenal gland. Part 2, explain the role of gland C in triggering an increase in heart rate in response to a sudden shock. So produces a hormone, adrenaline, travels in the blood, prepares the body for action, or you can say prepares the body for increased activity. So the adrenal gland produces the hormone adrenaline, which travels in the blood and prepares the body for increased activity by increasing the heart rate, increasing the pupil size, increasing the conversion of glycogen to glucose. So more glucose is available for you to in a situation of fight, flight, and fright situation. Now coming to question number 4, living organisms excrete carbon dioxide. Describe the meaning of the term excretion. Now, of course, I could give it to you in one sentence or I could give it to you in 3 sentences. Removal of waste products, 1 mark. Removal of toxic materials, 2nd mark. Removal of substance made by the body or made by metabolism. So if you said removal of toxic metabolic waste, well, you got your 3 marks. Well, it's only for 2 marks. But you could give it to me in 3 sentences or you could give it to me in one sentence and I would give you the marks for it. So, removal of toxic material or removal of waste products or removal of substance made by the body or removal of substance made by the process of metabolism. Now coming to part 2, name the process which produces carbon dioxide in living organisms and state how carbon dioxide is excreted by humans. So basically it is produced by respiration. So if you wrote respiration, you got a mark for that. And then how is it removed? It's breathed out, or you can say exhaled out. So it is breathed out or exhaled out, or you can say removed at the lungs or at the alveoli. Now coming to the B part of the question. The volume of carbon dioxide excreted varies over a 24-hour period and the factors that affect there are different for animals and plants. Suggest and explain reasons why the volume of carbon dioxide excreted varies for both animals and plants. Now, in animals' activities, like for instance, when you're exercising, you'll have more carbon dioxide as more respiration at some time when you're doing more exercise. In plants, respiration is constant, but during the daylight, there's more photosynthesis, so carbon dioxide is used for photosynthesis. So, activities varies, more physical activity, more respiration, so that will increase in carbon dioxide which is being produced by animals. In plants, respiration is a constant factor, but in daylight there is photosynthesis, so that carbon dioxide is going to be used for photosynthesis, and that is why it is going to be variable in plants. Then the last part, part C, explain the effect of an increasing concentration of carbon dioxide in the atmosphere. Now, of course, this acts as a greenhouse gas, traps heat energy, and this is going to result in global warming. This is going to result in climate change, and it's going to affect many animals and affect the land, of course, as well. So, increasing concentration of carbon dioxide in the atmosphere will result, it acts as a greenhouse gas, so this is going to cause global warming. And of course, you could give me any effect of global warming. I'm not saying that you can only give me this glaciers will melt and result in flooding, but you can give me any other effects of global warming as well. Question number 5. The castor oil plant is a flowering plant which produces seeds. The seeds have an additional structure called an oil body that contains oil. You can see this in the diagram. This is the testa. This is the main part of the seed. Figure 5.1 shows a castor oil seed with its oil body. Very interesting. The tester protects the seed contents from environmental damage. Name parts of the seed protected by the tester and describe their functions. Basically, you're going to give me the parts of the seed. So parts of the seed are what? Cotyledons. These are the food store which contains starch and they're stored for food stored for germination. So cotyledons, food or energy or starch stored for germination. Then embryo grows into the new plant. Then radicle grows into the root. I always tell you R radicle, R root and will absorb water. The plumule will grow into the shoot or the stem, which is going to have the leaves, and the leaves are needed for photosynthesis. So the cotyledons in the seed are the food store for germination. That's 2 marks. Cotyledons, 1 mark, food store for germination, 2nd mark. The embryo grows into a new plant, the radicle grows into root, and the plumule grows into the shoot, or you can say the stem. And basically, the leaves on the stem are needed for photosynthesis. Next part of the question says describe a laboratory test that can be used to show that the oil body contains oil. So, what will you do? You'll take that and you'll add ethanol, and then you add water, and a white emulsion will form. If the white emulsion forms, or you can say becomes white or cloudy or milky. So when you added ethanol, it's a clear solution. But then when you add water from the clear, it becomes a cloudy white emulsion. When you add ethanol, why is it clear? Because even if there are fats present in it, they will dissolve in the ethanol. It's an organic solvent. But then when you add water, then it separates out and a cloudy white emulsion forms. Part B of the question, when the fruits of the castor oil plant are ripe, they burst open, scattering the seeds onto the soil. Ants feed on the oil bodies, but not the rest of the seed. They either remove the oil bodies where the seeds have fallen, or they take the seeds and oil bodies back to their nest. Colonies of 5 different ant species A to E were supplied with the same number of castor oil seeds near to their nests. Figure 5.2 shows the results of this experiment into the behavior of these different ant species. Now it says oil body and seed taken to nest. In this situation, it's a dark line, and oil body only taken to the nest. So we have ant species and we have percentage taken to the nest. Now very carefully look at this graph and you can see that species A, let's look at it carefully. Species A, oil body and seed taken. So that's about 90%. If you look at here it's 90%. This is percentage taken to the nest. So 90% of ant species A took the oil body and the seed to their nest. So they helped in the dispersal of the seed. The B did not help at all, but B only took the oil body. Oil body does not contain the seed. C was helpful again. Why? Because it was nearly 84% and this was 90%. So 80% of the seeds were taken to the nest by ant species C. D was useless as well because very few, only this much, about 4%. And E was also useless because they took the oil body but they did not take the seed. Seed was only this portion. The seed was only little portion, this one here and this one here. B was totally useless because it only took the oil body, not the seed. Now let's look at the question. Only a small number of flowering plant species produce seeds with oil bodies. Suggest one advantage and one disadvantage to the castor oil plant of producing seeds with oil bodies. Now, of course, advantages increases seed dispersal. Disadvantages is it requires more energy because it will require more photosynthesis, more glucose, and then of course this glucose is going to be converted, is going to be used in lipid synthesis because it's going to have this lipid body or this oil body. So lipids need lots and lots of photosynthesis resulting in glucose and then resulting in respiration and the release of energy and the formation of fats or lipids. So increases seed dispersal. Advantage and disadvantages requires more energy for lipid synthesis. Next part is use information from figure 5.2 to explain how useful the different ant species are to the castor oil plant. Now I've just talked about that. If you look at it again, ant species A and C, ant species A and C are the most useful. And then you can give me a data code that 84% and 90%. That got you another mark for that. If you said 84% ant species C and 90% ant species A took away the oil body and the seed to their nest, then ant species B, D, and E, B, D, and E were all useless. Changar. I know you're very amused when I use the word changar. I use it a lot in the class as well. So B, D, and E is not useful as dispersal relies on the whole seed being removed and not just the oil body. You see, the whole seed needs to be dispersed. The oil body does not contain the cotyledons and the radicle and the plumule. The oil body is just an additional part to the seed. So A and C most useful. A is 90%, C is 84%, B, D, and E not useful. Dispersal relies on whole seeds being removed. Then part 3 was an easy question. The ants feed on the oil bodies and digest the oil. Name the products of this digestion: fatty acids and glycerol. Please do not write glycogen. This is a very important thing that you write the correct spelling of glycerol. And the last part of this question, ant species A is Solenopsis geminata. Figure 5.3 shows a diagram of S. geminata. S. geminata is an arthropod. The arthropods are classified into a number of groups. State the name of the group to which S. geminata belongs. Insect. And then they've asked you another question. State two visible features used to classify S. geminata in this group of arthropods. Well, it's basically three pairs of legs attached to the thorax, three sections to the body, head, thorax, and abdomen, and one pair of antennae. So, visible features which classify them as arthropods were six legs, or you can say three pairs of legs attached to the thorax, two antennae, and the body divided into head, thorax, and abdomen. And that was a 15-mark question, very long question. We will end the video here and continue the next few questions in another video. Thank you very much.